Hello, this is talk number 8 in a series of the book Pathology by Robbins and Cotran, 9th edition. Today we'll be discussing neoplasia. Cancer is the second leading cause of death in the United States. There's a broad spectrum of neoplastic cell types associated with all sites of the body. Neoplasia, by definition, is an autonomous, uncontrolled growth of tissue consisting of proliferating cells and surrounding and interspersed connective tissue, blood vessels, and inflammatory cells. Benign tumors grow locally and they can be fatal depending on the location. The suffix nomenclature is generally blank oma, and there are exceptions which exist. The prefix is generally the site of the tumor. Malignant tumors grow locally and discontinuously, meaning they metastasize, and they eventuate in death. Nomenclature, you have the tumor site and the category of neoplasm. Differentiation, by definition, is the degree to which a neoplasm resembles its normal counterpart. So we talk about undifferentiated, poorly differentiated, moderately to well differentiated, and well differentiated. And this refers to how much the tumor in question resembles normal tissue. And these are the examples here. You also have anaplastic tumors where there's high-grade nuclear morphology. Pleomorphism is an index of the neoplastic cell size and shape. Mitoses are an index of proliferation. They are not exclusively correlated to malignancy, although some malignant diseases have mitoses as a feature. Tumor cells can secrete hormones or other substances. We call these tumor markers. Dysplasia is a precursor pattern to neoplastic transformation, and these are premalignant cases. They are graded mild, moderate, and severe, or low or high-grade dysplasia. Malignant tumors, again, invade locally. They can metastasize to regional lymph nodes and or distant sites. The extent of local invasion and the presence of tumor-type metastases govern the decision to remove surgically. Metastatic behavior varies by tumor type. It can be organ-specific. Carcinomas spread by lymphatic channels to lymph nodes and then to organs. Sarcomas, or tumors of soft tissue and bone, they metastasize hematogenously via blood vessels. The epidemiology, some neoplasms are classified as epidemics, such as lung and AIDS-related tumors. Epidemics are, by definition, diseases that occur above anticipated rates of occurrence. These are data from 2014. They don't differ that much over time. We look at the estimated cancer incidence by site and sex for men. Melanoma of skin, lung, the highest number here is prostate at 20%. Miscellaneous categories are 20%. Lung is 14%. For women, the highest percentage is breast at 29%. 13% lung, colon 8% in women, and 6%, or rather 8% in men. These were data from 2014. Men and women with a very, very slight male predominance cancer incidence. For cancer deaths, the number is a bit lower. Men have a slightly higher percentage. For men, the highest number of percent of deaths in 2014 was lung at 28%, prostate at 10%, miscellaneous at 24%. For women, 26% lung, 15% breast, 23% miscellaneous. Genetics and environment contribute to cancer. Environment includes infectious agents, smoking, alcohol, diet, obesity, reproductive history, and exogenous carcinogens. The age is also a factor. Somatic mutations accumulate. These become non-repairable, and children can have neoplasms of certain types. The cofactors, there's chronic inflammation and mediators, reactive oxygen forms, and stem cell proliferations are all cofactors in neoplasia. Precursor lesions include histologic transformations. These lead to cancer in some cases. There are metaplasias, a change of cell type, and hyperplasias, which are increase of cell number. And immunodeficiency states are susceptible to oncogene viruses which cause cancer. Genetics are markers of increased risk. Often you need other predisposing conditions. The mutations can affect any of these gene types, such as growth-promoting proto-oncogenes, genes regulating apoptosis, growth-inhibiting tumor suppressor genes, and DNA repair genes. Mutations can occur in stepwise fashion, beginning with an initiating mutation and following by driver mutations. Mutations can cause loss of function and allow proliferation to continue unabated. Tumor doubling of neoplastic masses vary and they have a long lead time. The clinically evident neoplasm possesses a combination of genetic determinants permitting neoplastic growth, and alterations in DNA post-regulation structural changes, including methylation and histones, are contributory. The properties of neoplasia are as follows. Growth is autonomous, independent. They are insensitive to growth inhibition or growth inhibition factors. Metabolism is altered. They generally consume much glucose. Apoptosis is altered. There's increased replicative potential. In other words, the cells become immortal. Angiogenesis, there's a blood supply component. Many of these tumors develop their own distinctive blood supply. These tumors can invade locally and they can metastasize distantly, and they evade the host cell response. Oncoproteins act in proliferation, but they resist inhibiting signals. Proto-oncogenes code for accelerating factors. These are mainly receptor tyrosine kinase pathways in a host of cancers. Receptor tyrosine kinase comprises a group of factors involved in specific neoplasms. ERBB1 is epidermal growth factor receptor, also called EGFR. This is characteristic of lung adenocarcinoma. ERBB2 encodes HER2, which is amplified in some breast carcinomas, and there are RAS mutations. This is the most common type of proto-oncogene mutation, such as HRAS, KRAS, and NRAS. These are components of G proteins which are activated by receptor tyrosine kinases. Point mutations increase RAS activity and proliferation. Other protein kinases include BRAF, which activates transcription factors. Mutations increase function. A serine threonine kinase is present in many neoplasms. And also BCR-ABL is a tyrosine kinase translocation cofactor, which promotes activity seen in Burkitt's lymphoma and chronic myelogenous leukemia. This is the chronic myelogenous leukemia. You have normal proteins 9 and 22. There is a translocation that occurs, and you can see that part of this is transferred over to the gene. There's an uneven transference from 22 to 9. The BCR locus, you get a hybrid gene between ABL and BCR, and tyrosine kinase inhibitor is present. It does not act on this. There's activation of growth factor, whereas in a normal chromosome, you can also have this sort of translocation between 8 and 14. In Burkitt's lymphoma, you get the same sort of transference of MYC oncogene, and this creates increased MYC protein and increased expression of pro-growth genes here. There are other genetic alterations that occur in a host of leukemias and lymphomas, but these are two of the most notable ones. Their transcription factors, MYC oncogene activates cell growth genes. This is deregulated in cancer. They're tumor suppressor genes, and their inactivation by mutations creates cell growth. Sometimes you need inactivation of one allele if the other one is already defective. This is called the two-hit hypothesis. Examples are RB, TP53, and E-cadherin. RB binds transcription factors when they're hypophosphorylated. Neoplasm reduces RB function or creates loss of RB inhibition. TP53 is the most frequently mutated gene in human cancer. It performs many cell proliferation inhibitory functions through a variety of mechanisms, and as with RB, a previously affected allele through inheritance requires a second mutagenic hit on the other allele to create loss of function. P53 protein inhibits neoplastic transformation, including cell cycle arrest, cell senescence, repair, and apoptosis. E-cadherin is involved in proliferation cell response to epithelial injury. It requires cell-to-cell binding. Neoplasia causes loss of contact and allows for cell disadhesion and local invasion. And then there's a growth-promoting metabolic alteration. The Warburg effect is where neoplasms consume more glucose. This is the basis of positron emission tomography called PET scanning. Glucose with fluorescent markers localizes in neoplastic hotspots, and this is used commonly to ascertain the location of metastases or recurrence of disease. The factors of increased uptake, there's enhanced receptor tyrosine activity, tyrosine kinase activity. This is MYC-activated transcription factor, which supports glycolysis. There's the effect of neoplasia on apoptosis. This is a primary mechanism of disabling apoptosis in neoplasia, involving defects in the intrinsic mitochondria. Cells become immortalized. The P53 defect amplifies apoptosis decrease. And then there are other properties of neoplastic cells, immortalized cells, limited replicative potential, evasion of senescence, evasion of mitotic crises, which limit the pre-programmed number of cell divisions and characteristic of cell renewal through stem cell molecular similarity. Angiogenesis, tumors induce their own blood supplies. P53 inhibits overexpression of angiogenic promoters. This is inhibited by tumor. RAS upregulation in neoplasia induces angiogenesis. There's local invasion and metastases. The latter is extremely inefficient given the number of cells released. Local invasion requires extension into the extracellular matrix. There is extension through the basement membrane by tumor cells lacking adhesion factors. Degradation enzymes, the tumor cells are attached to extracellular matrix actin, and they are propelled forward to invade the interstitiums. This is an active process. This is an example of it. You have the cells in array. They loosen. You lose these intercellular junctions, desmosomes in skin, for example. There's degradation occurring here, attacks on the collagen, plasminogen activation, type 4 collagenase, and finally you get breakdown with the fibronectin and a migration invasion at this point, an autocrine mobility factor. Cells increase and they invade. Stromal cells and tumor cells interact to varying degrees. They result in tumor extension, cytokines, and growth factors. Metastases are a property of specific tumors that require a metastatic genetic component. Tumor oncogenes upregulate mesenchymal markers favorable for metastases. There's tumor evasion of the host response. Host cells recognize tumor cells as foreign. Tumors possess antigens which elicit an immune response. These are mutated gene products, for example, mutations of RAS, p53, and BCR-ABL. You also see antigenic responses to protein overproduction, viral oncogene products, and differentiated oncofetal products. Cell-mediated immunity, the most common defense is against neoplasia. This involves CD4 and CD8 cells, NK cells, and macrophages. Immunodeficient states lead to neoplasms. Tumors can escape immunosurveillance through formation of antigen-negative variants, loss of MHC molecules, and alterations of immunoregulatory pathways. Genomic instability includes aberrations which increase mutation rates. These acquire driver mutations, often involving mutations in DNA repair genes. The model for this is hereditary non-polyposis colon cancer syndrome and HNPCC. This is autosomal dominant. The genes affected carry out DNA mismatch repair, and this is what repairs single base point mutations. Microsatellite instability or defects in mismatch repair. These create errors in longer repeat sequences. 4 genes are involved, and you find this in 15% of sporadic colon cancers. Dysregulation of cancer-associated genes. These are often translocations determined in specific neoplasms. These are very characteristic of lymphoid and soft tissue neoplasms. A fusion protein usually has a deleterious effect on genomic regulation. Some oncogenes produce amplification, for example, HER2/neu in the breast. So selected examples of oncogenes created by translocations. Chronic myelogenous leukemia, again, we talked about BCL-ABL. Acute myeloid leukemia has 821, 1517 translocations. This is seen in M3 type. Burkitt lymphoma, 814 translocation. Again, P is the short arm, Q is long arm. So these are translocations between the long arm of 24 and the long arm of 32. The shorthand version is MIC 8Q24 and 14Q32. Mantle cell lymphoma, follicular lymphoma of the thyroid, Ewing sarcoma, generally a pediatric case, and prostatic adenocarcinoma additionally has some cases of genetic component. Not common, but it does occur. Non-coding RNAs in neoplasia, microRNAs mediate and inhibit messenger RNA translation. These are deleted or mutated to allow increased translation. There's a mechanism of incipient neoplasia, a multi-hit sequential mutations, for example, colon cancer. So we have a situation where you have a normal mucosal layer, submucosa muscularis propria, the lumen for the foodstuffs is here, and the remaining part of the intestinal lining is over here. Mucosa at risk, whether this is genetic or environmental. You get a transformation, methylation abnormalities, and you get a lesion occurring here, which can be exophytic or it can be sessile and flat. Sometimes the hard ones are the flat lesions, and they can develop carcinoma directly. Less commonly, though, with these everted villus lesions, as they progress in size, there's a great probability that part of this will occur as a neoplastic malignant transformation, and you end up putting this entire thing, the entire lesion, under glass, under microscopic review to find out where the cancer is, and more particularly, does it extend into the interstitium, Does it hit lymphatics? Once it goes to the submucosa, you have a problem in the staging. Depends on the depth of invasion as well as the presence or absence of metastases. These are genetic changes that occur in that process. So in tumor formation, we talked the colon model is a multi-hit process. Tumor formation has an initiating event and promoters of host or tumor origin. Some chemicals are direct acting. These are native chemicals, or indirect, which would require metabolic transformation. So among major chemical carcinogens, we have some which are clearly problematic. These are in large classes. Alkylating agents, acylating agents. You have metabolic, these are direct acting carcinogens here. You have ones that require metabolic activation, such as polycyclic and heterocyclic aromatic hydrocarbons. Benzoyl-pyrene, for example, is the active component in cigarette smoke. They're aromatic amines, amides, azodies, benzidine. There are natural plant and microbial products, aflatoxin B1, which causes massive liver damage. And you have a miscellaneous category, including vinyl chloride, which is problematic in the urinary bladder, and insecticides, which are correlated with hematologic neoplasia. Polychlorinated biphenyls. These fall in the category of toxicology. Non-chemical carcinogens. UV radiation has 3 spectra: UVA, UVB, UVC. UVB is carcinogenic. UVC is filtered out by the ozone layer. Ultraviolet light causes pyrimidine dimers from thymidine. These are normally repaired. Ionizing radiation includes X-rays, beta particles, and alpha particles. They cause chromosome breakage. These are dose and energy dependent. Viruses of the RNA type also are carcinogenic. HTLV1 is human T-cell leukemia virus. This is similar to HIV, but has a different geographical distribution, found in South America, Africa, and Japan. Among DNA viruses, which are carcinogenic, the human papillomavirus has 70 serotypes with differing disease courses. The benign lesions include plantar and cutaneous warts. Malignant lesions are found predominantly in the uterine cervix, oropharynx. These are sexually transmitted. The DNA is integrated randomly and forms clonal proliferations. Epstein-Barr virus infects B cells. This is correlated with Burkitt's lymphoma, which is a B cell proliferation seen in Africa and Papua New Guinea. These act as a polyclonal mitogen, and they allow for an 814 translocation, as described previously. Hepatitis B and hepatitis C are respectively DNA and RNA viruses. These are immunologically mediated inflammation and cell death, allowing for mutagenicity, a lengthy process. Helicobacter pylori is a gastric microbe. This causes ulcers and gastritis. There's inflammation-induced mutagenesis with a risk of B-cell lymphoma. Cancer effects. These have local growth and mass effect. Rare paraneoplastic hormone secretions. Cachexia is cancer-induced starvation, seen usually in terminal phase. Coagulopathies can occur. The grading is the histologic appearance of the tumor. Staging is an index which encompasses the primary tumor size, the extent of local spread. So the tumor size and local spread. Local spread is determined by T, tumor, no metastases, N, and presence of distant metastases, M. These are stages 1 to 4 with the numerical subscript based on the subcategories. For example, a T2, N1B, M0 tumor. So pathologic means of diagnoses are include in the field of anatomic pathology, fine needle aspiration, where a needle is used to remove cells which are spread, fixed, and assessed under the microscope or through immunocytochemical or histochemical means. Cytology smears can be gynecologic smears or non-gynecologic. There are small biopsies, which are either incisional, meaning you take a piece of the tumor or the piece of the lesion, which is uniform, or excisional, meaning you are removing the lesion with a rim of normal tissue around it. They can be localized or en bloc resections. Ancillary methods include flow cytometry, immunohistochemistry, histochemistry, and cytogenetics. The clinical pathology branch results from hematological, clinical, chemical, and microbiological fluid samples, assessing for tumor markers where applicable. To this, we also add the area of blood banking and the components derived thereof. So the last thing that needs to be shown on this lecture is tumor markers. So certain types of diseases produce hormones, they produce oncofetal antigens, they produce isoenzymes, specific proteins, mucins and other glycoproteins, and DNA markers. So you can use the DNA markers as one set of studies, or you can chemically and quantitatively assess for these other entities, such as human chorionic gonadotrophin found in trophoblastic tumors, testicular tumors of certain types, calcitonin seen in certain types of thyroid tumor, ectopic hormones, alpha-fetoprotein seen in some forms of liver cancer, carcinoembryonic antigen seen in colon cancer and a few other diseases, prostatic acid phosphatase in prostate, CA125 in ovarian, CA19-9 in colon, and 15-3 in breast cancer. These are all quantitative markers. These are called tumor markers, the presence of which does not guarantee a tumor, but in the presence of a prior diagnosis of a tumor, increases or changes in concentration of one of the tumor markers can be indicative of a recurrence of the disease. And that's it. Thank you very much.